All right, so I'm Jessica. Welcome to PHAVES 9. We are going to start with a little bit of announcements, and then I'm going to introduce our speaker, Rodrigo, and then we're going to get going. So just a couple announcements. It's almost World Phage Week, so this is kind of, I've seen it around on Twitter, and there's a hashtag World Phage Week, October 22nd to 28th. So that's led a lot of people to start hosting more virtual events related to phage. So just wanted to let the people know that the Ibadan bacteriophage research team is hosting talks on the 26th and 27th. And that's all about early career research in phage studies. And so I think they're not science talks. I think they're more careers focused, but maybe with a mix of science. With Urmi Bajpai, Nadi Namika, Stephanie Lynch, our own PHAVES volunteer will be speaking, and Dr. Sabrina Green. And then we have Proteon Pharma on October 28th, talking about E. coli phage application in poultry. And then our next PHAVES is Dr. Paul Jaschke from Macquarie University in Australia, and that's November 10th/11th if you're in Australia. So that's our announcements. And as far as today goes, the way that we'll go is similar if you've been here before. We'll have Rodrigo talk for about 20 minutes, and you can type your questions into the chat. You can use the chat for anything, like introducing yourself. Hi from Canada, for instance. That's great. And then also if you have tech problems or you hearing people, feel free to just message us in the chat as well for that. And I'll be either calling on people if they would like to unmute during the question period. That's Rodrigo's favorite way of doing things. So I'll kind of call on you if I pick your question, and then you can optionally ask it yourself. And we're going to go ahead with that kind of method this time. So let's introduce our speaker, Dr. Rodrigo Ibarra-Chavez. He is a postdoc at the University of Copenhagen in Denmark with Professor Søren Sørensen. And he did his PhD at University of Glasgow with Professor Jose Penades, and he did a PhD in biomedical engineering. And so he's studying these PICIs, PICI, that's the fancy acronym for them.
Acronym,
yeah, the family of mobile genetic elements that actually parasitize prophages. So if you get this question from your family members as a phage researcher like I do, it's like, oh, we study viruses that kill bacteria. Well, does anything kill those viruses? This is that thing. So Rodrigo's going to talk about those and discuss his research today. So I'm going to turn it over to him now.
Thank you. I'm just going to get, yeah. Can you see my cursor? So just in case I can, highlight.
Yes,
okay.
I can see it.
Yeah, so I'm going to talk about phage-inducible chromosomal islands. And I was telling Jessica that, some people, when they talk about phages, we normally say, yeah, they are the hijackers of cells. Well, in this case, these are the hijackers of the hijackers, which is a lovely term we use. And there's a very nice paper that talks about that. So yeah, as you know, mobile genetic elements are very important. They drive horizontal gene transfer. And essentially, that's the major way by which bacteria acquires the genes that enable a rapid adaptation and evolution to very specific environments. So in this case, bacteriophages take a big role in horizontal gene transfer because they not only disseminate themselves and maybe they can carry some, beneficial genes for the bacteria cell, they also can mobilize other elements, other mobile genetic elements, such as plasmids, even regions of the chromosome, and in this case, also the phage-inducible chromosomal islands. So just to talk more about horizontal gene transfer, now there are three mechanisms that we know, and we have worked with it, and it's incredible how these mechanisms can actually, transfer genes into other cells. So as I said, this is the major route by how bacteria acquires new genes into its arsenal and adapt better to other environments. And which type of genes you're probably thinking is virulence factors, antitoxins, biofilm formation genes, and most importantly, antibiotic resistant genes, which is a very hot topic at the moment. And these mechanisms are generalized, specialized, and lateral transduction. I'm not gonna explain them in detail, but if anyone wants to know more about them, I really recommend you go to this review, and then also the paper that lateral transduction was described on from my friend Nuria and John Chen. And in that regards, the life cycle of the phage is very important. So for all the phage community, I'm now speaking only about temperate phages. I haven't had the pleasure of working with lytic phages yet, but these are mainly lysogenic phages, like phages that can go dormant in the bacterial cell. So as you can see here, the cycle, I'm not going to go through it because I hope you guys know it. For the public that doesn't know, I'm sorry, I need to keep this talk interesting for most of the phage community. But one of the more important aspects about phages is that they have given us most of the biological tools, like the molecular biology that we are commonly use every day in the labs. It comes from a lot of research in phages. And if you want to know more about that, I really recommend that book, The Phage Lambda, A Genetic Switch. You can buy it on Amazon. It's pretty easy. And saying that, you know, I will consider them as the natural engineers of life. They are able to, you know, edit bacteria and then, you know, as we normally say, copy and paste the genetic sequences. So, and they also evolve very quickly. So there are different tools that we've developed with them. There's genetic regulation, which, you know, we can play with transcriptional regulators. There's genetic methods for, you know, rebooting phages, but also to accelerate evolution of promoters or even proteins. There's more about the structural part of it. So like phage capsids to develop nanocarriers, to develop vaccines, and phage display, which is a very important application, for example, nowadays to maybe test COVID. And that we use, like, you know, the peptides to actually detect antibodies or actually the virus per se. And interestingly, most of the genetic switches and most of the applications that we or that have been developed is mainly of a couple of phages. And in the world, there's like 10 to the 39 phages. Like, it's a vast world. Like, there's more phages than bacteria. So what else is there? And I'm going to show you what else is there. So phage-inducible chromosomal islands, that's another thing that is in there. And we know, as I said before, they are the parasites of the parasite. And they're very highly evolved. They're very intelligent. They're very, I don't want to say intelligent, but they know what they're doing. And they ensure the dissemination. So they protect their host, but at the same time, they love to have the phage to actually reproduce, so they can go into other cells and infect other cells and then transfer other things. They were initially described in Staphylococcus aureus, and these were named Staphylococcal pathogenicity islands. Mainly one, SAPI1. SAPI1 is the one that carries the toxic shock syndrome toxin. And that's how it was described. At the beginning, many people thought these guys were defective phages. They're genomic islands. And at the beginning, they didn't see to, you know, have any instance of actually, you know, producing capsids or going on their own. But actually, they reshape the capsids in certain types. And you can see here in this image that you can have a phage, but also a PICI. And in this case, this SAPI is able to reshape and ensure that only the genome, which is smaller, gets into the capsid. I'm gonna talk more about the size of the genome. The genomic organization and regulation is very similar to what the phage looks like, and that's why a lot of people thought at the beginning these are probably defective, because they look like phages, but they're not entirely like phages. You have missing parts, and then people said, nah, that's a defective phage. It doesn't really work. But then the most interesting thing now is that we can see that they're widespread amongst bacteria. And this is pretty cool, because what else are they doing? Are they only for mobilizing things, or are they also doing another role in bacteria? So what is the modus vivendi? How do they normally live? How do they normally go about in life. So you can have two cycles, and you can have one where you have already a prophage in the chromosome of the bacteria, and then by a SOS response, you induce the helper phage. We normally call them helper phage, the ones that are actually inducing the PICI. So if you induce the helper phage, that will cause a reaction to go and induce the PICI, and then the PICI will start its cycle of replication and then also interference to actually ensure that the DNA of the PICI goes into the capsids of that the phage is developing. And also you have the other a scenario where you have the helper phage infecting the SAPI, because the helper phage wants to reproduce. The PICI detects that and then also exploits that. And then ultimately you end up with a lysate, which has a number of phage particles and a number of PICI particles. And then those PICI particles can go into another phage, into another cell, infect, and then integrate themselves and then stay there for how long they want until another helper phage induces them. Sorry. So as I said, they normally exploit the helper phage. So these are the ones that actively induce them, and that induction causes a very high transfer of the PICI. But then we've seen also that they are able to exploit a wide range of phages. So it doesn't have to be only the helper phage, because they have other interference and other modules that interact with them. They're actually able to transfer themselves at lower frequency as what we could say like a normal generalized transduction, for example. But they also, we can see that they also driving the dissemination of chromosomal genes, because ultimately they are located in the chromosome, and normally they are even in different sites. So that's a thing you can look at in the paper of lateral. Some of the attachment sites for at least the SAPIs, the ones that we identify, Nuria has seen that, it's very specific. So that's a very interesting thing, because for example, genome engineering, you have another attachment site where you can put things in. So the genomic structure is very similar as the phages. You will have, flanking regions in the genome, and then the attachment sites, and then you will have normally a module that regulates the lysogenic cycle. So that would be, for example, in this case, what you see here, STL and STR, that would be C1 and CRO-like switches, like in Lambda. And then you'll have the replication module. And then commonly after that, it comes the packaging signal. I'm not going to touch much into it, but some phages, temperate phages, they can pack into a pack type mechanism or a COS type mechanism. A COS being, if the phage has 40 kilobases, the number of genes and the number of DNA that is able to pack has to be, between those COS signals. And with a pack one, it's normally detects one pack and then cuts whenever the capsid is completely full. So some of them can drive, this kind of transduction that actually gets other genes into the capsid. And then after that, we see modules that are able to interfere to redirect the packaging for the PICI DNA, and some of the toxins, some of these factors that they spread. And commonly we try to, for example, myself, that I engineer them, we normally try to delete those and then put things that we can track their mobilization, or even for other applications that I'm going to show later. If you want to know more about interference, I recommend Alfred's review about the PICI interference mechanisms. And also there's another type of elements called PLEs, PICI-like elements, in Vibrio cholera, and these are very interesting, and they have a very different lifestyle, but somehow they're very related. Like it's a different interference, and it's lovely to see what they're doing there. But what are the other roles of the PICI? So is there anything else rather than just spreading antibiotic resistant and virulence factors? And there's some PICIs that actually we don't see we cannot see anything on them. So is there another role? Is there maybe a way of protecting the cell against the phages? and actually, not completely abolish the phage, but still enable the phage to reproduce and ensure that this kind of protection passes on to other generations. So it's a thing we're working on. But for example, the model, the staphylococcal PICIs, the SAPIs, the most common ones will carry 3 types of interference. Would be the packaging interference, the packaging redirection. It would be late interference with the transcription of the lysis of the phage. So it kind of delays the lysis of the cell. And then also the capsid redirection. So instead of having big capsids that enables packaging of all the DNA of the phage, it reshapes it, and then it's perfect for the size of the PICI. And the PICIs are normally a third of the size. So about 15 kilobase, 17 kilobase pairs. So they hijack, some of them hijack the terminases or interfere with the phage DNA. So it completely blocks the phage DNA and then ensures that most of the PICI DNA gets in the capsids. And then, as I said, they interfere with the slowing down the lysis, and they redirect the size of the capsids. And it's interesting because for certain applications that would be useful, meaning that you have a smaller capsid, maybe you can have like more, less space actually bouncing each other, or the fact that you can pack more DNA, and then you want the bigger capsids. And that's a thing we actually did in my paper. But so far, as a take-home message, I want you to see what are the advantages of being a PICI. I normally call them like a Trojan horse, because what you're seeing is a phage, but what is inside is completely different. So it's a very nice analogy, and they gain the characteristics, the physical characteristics of a phage. So when we talk about receptors and tail fibers, those are basically the ones for the phage. And then they manipulate the life cycle of the phage to promote their spread into other cells, and we think also to protect some cells. And then the very nice thing is that it's a third of the genome of the size, so you can add things into them so they can carry it to other cells. And because they evolve they're very highly evolved mechanisms, they are able to swap things, and they're able to adapt as well. So we see these changes in like, for example, clinical strains, and it's very interesting. And we're trying to see what does that mean ecologically and environmentally. And so far, very interesting for the enthusiast of CRISPR. So far, we haven't detected an anti-PICI system, and you can genetically engineer them. And that's what I'm going to show you next. So one of the very first things we come up with was, how can we utilize the packaging interference to redirect the packaging of a plasmid that, for example, carries CRISPR-Cas. So we use Cas9 and Cas13a. My collaborator Kiga, who's working in the Jichi University in Japan, he brought some of the Cas plasmids, and we put some guide RNAs against carbapenem-resistant genes, and we added that into it, the cause signal, so the packaging signal, and also the RPP, which is the packaging redirection protein from the gram-negative PICI in E. coli. And then we mutated the phage, so the phage wasn't enabled to be packed, and then we basically use it to produce the capsids, and then the PICI will just, the parts of the PICI will enable a higher encapsulation of the plasmid, and then a transfer, ensure the transfer into the other cells. And we target these genes, and we saw we can target genes in the chromosome, but also genes in the plasmid. And comparing both types, the Cas13 and the Cas9, you can come up with different ideas and different applications. For example, if you just want to cure a plasmid and you still want to, keep the bacteria cell, then the Cas9 would be very useful because it will just get rid of the plasmid. The DNA will just, the nucleus will just degrade the DNA. And in case of the Cas13, because how it interacts and the collateral damage that causes in the cell, it kills even if the sequence is in a plasmid. So the idea that we came up with was you can also use this technology for detecting bacteria. So in this case, for example, if you want to detect that a bacteria has an antibiotic resistance, but then maybe plasmid or sequencing is not the best solution. In this case, this type of scenario will give you because it's not growing, this bacteria has the antibiotic resistant gene that you ultimately don't want to continue culturing. And then we come up with the rebooting. So how can we genetically engineer them better? For example, normally, doing genetic engineering in E. coli is easy, it's fairly easy. But in some other organisms like Staphylococcus, Enterococcus, is a bit more complicated and it's a bit more tedious. So how can you actually, engineer big chunks of the PICI or the phage and use them in your advantage? So very similar to the application that Ando developed and also Kirchner in the L-form method. We use the yeast assembly method. So we use PCR products to amplify the genome of the PICI, and we amplify it not as if it was in the capsid. It was as if it was in the chromosome. So we can ensure that if it has flanking regions and also the integrase that is very efficient, it helps us to recombine into the chromosome and ensure that it actually integrates into the proper attachment site. So it's a fairly easy method. You can check it out in the paper in biodesign, and I'm keep working on this. Like we want to expand the arsenal of how you can reboot and engineer PICIs into other organisms. And in this case, we repurpose one of the SAPI, so another PICI, for delivering synthetic genes as well. So not Cas9, and also in the case of GFP and beta-gal. So similarly to the previous approach, we did a defective phage that was not able to pack its DNA into the capsids. But then the PICI, with the packaging signal, we were able to integrate the synthetic modules. So in this case, CRISPR-Cas, GFP, and then because it has a smaller size of DNA, you can actually play with that. So, for example, for this type of PICIs, you can have 27 kilobase pairs of synthetic DNA that you can actually put and then pack into the capsids. So yeah, you drive the phage, that you induce the phage, and the phage starts reproducing the capsids, and then the PICI ensures that its DNA goes into it, and you end up with a very high titer of PICI particles, no phage particles, which that's nice if you don't want to, encourage the dissemination of other type of traits, or actually, actually kill your cell, because that's a very interesting point for diagnostics. This is an application that can be used for processing other reprocessing or after processing the sample. So 2 approaches. One of them is for killing the target pathogen, and the other one for detection. And basically, as I said, you can have a very sequence-specific killing by using CRISPR-Cas, or you can also identify a bacteria and also enable a sequential processing. So if you want to, check if it has antibiotic resistant genes or nasty traits, then you can then, come up with better solutions to treat the infection. So in the paper, we developed one which was a Cas induced by cadmium, and we were targeting a ribosomal regulator. And then also we did one against the McKay in Staph aureus, and we actually test that against clinical strains. And as many things happen with Cas and phages, like I don't think it's one bullet that you're able to shoot if you really want to ensure a complete eradication of an infection. I think you need to have the synergy of different things, maybe something that targets virulence factor and then something that actually kills the cell. So you can have an antibiotic and then maybe a PICI delivering a CRISPR system. And then, as I said, you can also have the GFP for detection. But this is just the tip of the iceberg. Like, this is the only real applications that have been developed with PICIs. And we're working on what else can we do and what else is needed to keep doing these very interesting applications. So now we're studying the regulation and the complexity of the PICIs, and also with other cells, because even the clinical strains, some of them carry more than one. So what is going on there? Is there any crosstalk? Is there anything more that they can, sense or do? And as I said, we're working on better platforms to engineer them, to design them, but also there's no real identifiers. We need a really nice bioinformatic program for that. And then how can we repurpose the PICIs? So one of the ideas is like, can we use them as anti-phage mechanisms? Like, can we manipulate, a population to actually, if there's a phage that carries a toxin, then try to avoid that. Or if you want to be very specific, for example, in the microbiome, then target a very specific set of pathogens, but not have the other, harming the other nice population of bacteria that you have in the microbiome. And in that sense, now people has seen that temperate phages actually have a very important role in the microbiome. So what's the ecological impact of it? But also, what is the ecological impact of PICIs? If PICIs are already widespread, what is happening in there? And it's very interesting. So as I said, it's just the tip of the iceberg. And I hope you guys like the talk. Obviously, all this work wouldn't be possible if it wasn't for Jose and all the collaborators and all the people that I've worked with, Andreas, Nuria, Kiga, Professor Cui, and also Inigo and Pedro. Pedro came to our lab and did a lot of work with me. He shared with me the guide RNAs against RSA. So yeah, that was great to have him. And it's been a nice time in Glasgow. And now I'm passed to other things. And we're developing very interesting applications here. And I'm so happy that, Orvish and Soren accepted me to come here. And I hope you guys stay in contact, and we're open, if you want to collaborate, if you need any kind of help, we're here. And I'm sure also the people with Jose, they're also very approachable. So please do contact us. And lastly, this thing wouldn't be possible if it wasn't for a friend. Sadly, he passed away last week, and he was a lecturer in university. So for some of us, we wouldn't be here if it wasn't for him, because he gave us lectures about genetic and molecular biology. So it was a very sad thing to hear. But yeah, it's in his memory, and it wouldn't be possible if he wasn't there. Thank you.
Thank you so much, Rodrigo. That was great. That was really interesting and really lovely dedication at the end. We are with you, the phage community, and we keep losing phage scientists. We also lost Dr. Richard Callender this month too. So yeah, all the more reason to keep going forward. And so, okay, we're going to do our question and answer period now. So we're going to kind of experiment a little. Normally I read the questions. I'm going to call on people who've written in questions. And if you would like to unmute yourself and ask your question, I'll give you a few seconds to do that. And if we don't hear anything, I'll just move on to the next person. So if you have asked a question and you do want to, be ready with your unmute and you'll get your chance. And that way, Rodrigo will be able to see who it is who's talking. That's kind of the main goal of it, okay? So let me go ahead. Atif Khan, you've asked a few, so why don't you pick your favorite question if you would like to ask Rodrigo. Maybe this won't go so streamlined, but we'll see. Okay, we're gonna let Atif see if he's coming back. I guess this is how radio hosts feel. All right, I'm gonna pick Anna Dragos. Would you like to ask your question?
Hi, Rodrigo, and
Hi, Anna.
all best from DTU, Lyngby. Yeah, my question is, do you think these PICIs can recombine with helper phages, and do you see any mosaicism in these elements? What do you think about it?
With helper phages, no. We haven't seen that, at least the ones that we've studied. And also, I guess one of the reasons is because they don't share the same attachment sites. So yeah, we haven't really seen sequence or genes that they share with other phages, but we have seen it between them. So as you can have mosaicism between phages, apparently PICIs have the same. So,
Okay.
but between PICI and phage, no, not so far. No, not that I'm aware of.
Thanks. I will get in touch with you for sure later.
Okay. No, but have you seen that?
We will talk. I have some system in Bacillus that might be, maybe it's a suspect picky, but we don't know. Okay.
Yeah, that'd be great.
Okay. Awesome. I'm muting myself.
Thank you.
Thank you. Okay, it works great. It's perfect. All right, Newton Mishodingi, if you would like to ask one of your questions, here's your chance. And after that, oh, I hear something. Is that you, Newton?
Yes, sure. I need to ask. I mean, the presenter, I think I have addressed it on the chat. What is the housekeeping gene in the phage? In that, we can take the example of PICIs genome or any other phage. The housekeeping gene.
The housekeeping gene, do you mean by like the C1 or the CRO or like a gene that we always see in the PICIs?
Yes, some people, when they want to use some bioinformatic pipeline, they do consider that the genome of phages is not really huge. So they can go for full whole genome sequencing. But this is not really, maybe it's somehow it's quite expensive, and it is not normally recommended when you have many samples. So we are trying to find out whether there is any housekeeping gene. And yes, so that
I mean, I guess it will change, but if I think it's the integrase, one of them, because they would be in the genome, so that would be they will need that for integrating. We've seen that the terminases seems to be quite conserved for the ones that use terminases, because there are PICIs that don't have the terminase. But yeah, normally we go by the modules, like you have the integrating and lysogeny regulation module, and then the replication with the signal, the cause or the pack, and then you will have the packaging module, which is changes from one to another. I would say the more conserved ones would be the lysogeny and the replication.
Thank you. Awesome. Let's move on to Pranav and or Apurva from Vitalis. I like your question about abundance. If you would like to ask it, go ahead.
Yeah.
Hi, Rodrigo. Thank you for that really, really interesting talk. This was the first time that I'd ever heard about PICIs. My question is, what is the abundance of PICIs as compared to phages? And I do have another question as well, if I'm allowed to ask.
Sure.
We still don't know. We still don't know. And that's the thing. We want to know, because there are genomic islands that obviously they act very differently. So we need to come up with better bioinformatic tools to actually know how much there is in. I don't think anyone has looked yet to like consortia of samples or like what is the PICI population. But actually, that's why I'm here in Copenhagen. That's why I'm working with Urvish, because they know how to do this here. So hopefully we'll do it. Yeah. Well, actually.
And my next question was about, do bacteria recognize PICIs better than phages, or do they recognize PICIs as being symbiotic with them as opposed to phages being predatory towards them? Is that something that you've noticed in your research as well?
I mean, it's actually, we know that the bacteria likes the PICIs because they protect them from the phage. Okay. But then we also see that in a circumstance that another bacteria might come onto play, or a harsh environment, then the PICI will actually like the phage to get it out. So it's a very interesting dynamic because, for example, other systems, they will completely abolish the phage. And the PICIs don't do that. Like they diminish probably 3 logs, maybe sometimes 4 logs the titer of the phage. And in some cases, you can see like a very nice interference, but it's not complete abolish. So they protect the bacteria against the predator, but sometimes they also carry biofilm producing genes. They carry toxins for certain diseases. But for example, if I have an example, the SAPI-BOV1, which is normally common in bovine strains, if it's not necessarily, it will just spit it out. Like the cell will just discard it. So it's not super stable, and we see that some PICIs actually can just go out of the cell. So there is indeed like a synergy, or I think we think it's a bit more complex.
Okay, thank you.
Awesome. Okay, the next questioner, I want to pick Adriana Hernandez because she just finished her PhD, I believe, this week. So you earn yourself a question if you would like to ask it, Adriana. She might have stepped. Oh, there she is.
There she is.
Hey, congratulations. It was a great talk. And I should confess, we've read a bunch of your SAPI papers in Journal Club. So
I know, because Rylan was in Glasgow a few years ago. Like he comes to
and
visit.
did he mention that we read a bunch of your papers in Journal Club?
No, he mentioned that he was gonna make that, but yeah.
Okay, so yeah, we have. And thank you,
Nice.
Jess. So is there any known interaction with these SAPIs and virulent phages?
Not that we've seen yet. At least in my awareness. But for example, the PLEs, the PICI-like elements in Vibrio, those interact against a lytic phage, a virulent phage. So if we might dig up a bit more, maybe not in staph, but other species, probably those PICIs actually do that. I guess it also depends, because the environments are very distinct. Like if you compare staph and a Vibrio or even E. coli, very distinct ways of interaction, and even how they communicate the cells between each other. So it's also interesting to see that, for example, staph doesn't have many CRISPRs. So for example, Pseudomonas, I've looked into PICIs with Pseudomonas, and there's a few. There's no very much on, but there's a lot of CRISPR in it. So I guess the bacteria knows what it needs and then evolves with it. So I wouldn't be surprised if there's a PICI that can actually hijack a virulent phage.
If I may ask another question.
Sure.
So I found very interesting the use of PICIs to modify bacteria. How do you envision that being done on a widespread of bacteria?
I think because we've seen that the integrases are a bit more promiscuous. And that was a thing we were a bit afraid when we were putting it into the yeast. We didn't want aberrant integrations. But at least when I was working with it and then after rebooting it, we didn't see that. So that was very nice. And in a way, I think, as I mentioned, the fact that they have completely different attachment sites, it's a super cool thing because you have another option to integrate things and to actually, put big chunks of DNA probably in a cell. So I think it's a great potential there, and I would really like to, keep pushing for. I just today, Rob and Pablo, they published a very nice review about, utilizing phages from other sources and other hosts, and doing these genome engineering. So I think it applies the same. Like use the PICIs as well. Like there's so much arsenal at the moment that it really needs to be exploited.
Thank you so much, Rodrigo. It was really good.
Adriana. Thank you.
Awesome. The next, we're circling back around to Atif. You can go ahead and ask your question if you would like.
Yeah, it was a very nice talk, Rodrigo.
Thank you.
I have one question, like who is more responsible for this spreading of antibiotic resistant genes in the environment, bacterial conjugation or PICIs?
I mean, depends, I guess, in the bacteria. Like, I think in gram-positive, some of them carry some antibiotic resistant genes, but they mainly carry virulence factors and biofilm stuff. We've seen that, for example, in E. coli, some of them might also transfer antibiotic, but it really depends. And a conjugation, I haven't really dig into that world. It's a completely, different area of horizontal gene transfer. I mean, I guess it's both are fantastic ways of sharing genetic information. It's just that, in conjugation, you need that contact. And here the phages phages are very stable in the environment. One of my first experiments was actually, evaluating how long can you have a lysate of phages and PICIs, and you can have them for months. So that's a pretty cool thing.
Super cool.
I have one more question.
Sure.
Yeah.
What is the basic difference between PICIs and prophages?
What is the what?
PICIs and prophages. What is the basic difference?
Well, they can produce particles on themselves.
Oh, okay.
So normally some of them carry some capsid formation or some structural genes for the capsid, but you will never have. So if you isolate a PICI, or like if you come up with a synthetic PICI, you will never be able to produce a particle on itself because it needs the helper phage.
Awesome. Very cool. I'm going to say, how about 2 more questions, and then we're going to do a breakout room. So I'm going to ask Sabrina Green if you would like to ask yours, and then after that, it will be Adam Ostrowski after her. Okay, so go ahead, Sabrina, if you would like to ask your question.
Someone answered my question already, so I think I'm okay.
Awesome. Okay. Adam.
Hi, Rodrigo. Greetings from Misty, Scotland. I was wondering, from putting the PICIs to a good use, how is using that sort of approach different from the well-established engineering of phages to carry, or CRISPRs, toxins, marker genes, etc. Versus using PICIs? Is it better? Is it different?
The biggest difference is that, the phage, it has like a very narrow size. Like you have very few DNA base pairs that you can manipulate if you want to carry the phage. That's one example. If you decide to have,
just use a phage capsid, but it's what the phage is carrying. It doesn't actually contain the phage genome.
So in the case you want to, for example, deliver a plasmid sometimes can be, the restriction modification systems can kill them, can destroy them. Sometimes the episomal, plasmid simply just doesn't carry on the cells. And the advantage is that with the PICIs, you have these 27 kilobase per span of adapting modules that you want. Also, you ensure the integration into your host. And there's no spontaneous induction. There's no spontaneous, lysis. So as I said, if you want some sequential processing to keep asking questions about the pathogen that you isolated, it's a better way to do it. And I think one of the beautiful thing of them is that, they have these genes that repurpose phages. So you can come up with very nice ideas. And that's some of the stuff that we're working in.
Cool, thank you.
Awesome. Okay, we do have one last question. How about from Ursa? Would you like to ask your question?
Yeah, sure. So if I get it right, that PICIs enclose themselves in a prophage capsid. And so the specificity on which bacteria they can infect is totally dependent on which prophage capsid they enclose themselves into?
Yes.
Okay.
But the nice thing is that they can encapsulate in different phages.
Okay, so it doesn't.
It's not only one phage that they can manipulate.
Of course.
So that's pretty nice. And actually, something that Jose and other people in the lab saw is that there's something called silent transfer. So and I really hope you guys are on board of this. The fact that a phage goes and infects a cell and then reproduces, and you see the phage plaques, that's one thing. But then there's a completely different aspect about if the phage infects the DNA and stays there. You might not be able to reproduce, or you might not be able to produce, viable particles and lyse the cell, but it doesn't mean that the DNA didn't go in. So we call this silent transfer. And that's how they detected that the Speci were actually passing from Staphylococcus to Listeria, for example. So it's a very interesting thing that, maybe there's something there that we're missing, because we studied, it's so nice to see the phage plaques and the interference, but maybe there's other ways that we can start studying this infection and these interactions, because we might be missing this. And if we see it in Speci and other PICIs, might be also happening with phages.